Thank you very much, everyone, for attending our Tech Talk today. We are absolutely honored and very excited today to have Dr. Kneil from University of Delaware to give us a talk on the role of wastewater epidemiology and how it has been used during the COVID pandemic. This is wonderful and fascinating. But before I introduce our speaker, let me go over some housekeeping issues. So as always, we will continue extending CME credits, and if you did not register prior to today's talk, you can still obtain CME code information from Deborah Rees, and I'll give her email address in the chat. Our next ID series presentation on October 28th is from Christine Brown from Christiana Care, and she will talk about getting started with Teachback because October is the health literacy month. On November 4th, we'll have another tech talk from Mary Jo McDuffie from University of Delaware, who will talk about Medicaid data for research, so you really do not want to miss these two presentations. And on November 11th, we'll have another ID series from Katie Fitzpatrick, also from University of Delaware. So these are really exciting presentations that are coming up. So again, I'm really honored to have Dr. Kneil here to give us a talk. Dr. Kneil is professor and associate chair of the Animal and Food Sciences Department at University of Delaware, as well as the director of the Center for Environmental and Wastewater-Based Epidemiological Research. He is also the current past president for the International Association for Food Protection, and she obtained a PhD in food science with a focus in food microbiology from Virginia Polytechnic Institute and State University in Blacksburg, Virginia. She has received multiple honors, last one in 2020, where she received the Excellence in Research Award from University of Delaware College of Agriculture and Natural Resources. She also has received numerous grants from USDA and several foundations. She has multiple peer-reviewed publications as well as textbooks and book chapters. She has been invited nationally and all over the world to speak about food safety, and she is a teacher, a mentor, and has served on multiple dissertation committees. So Dr. Kneil, again, it is really a pleasure to have you here to talk about your research. Thank you so much.
Oh, well, thank you. That is such a lovely introduction. And it really is an honor to be here with you, and especially with the exciting lineup of speakers that you have. So it's definitely an honor to be a part of this and to be able to talk to you today about some of the past, present, and future use of wastewater-based epidemiology. So some of you may be familiar with this setup here that's on the slide to the right. This is a composite water sampler, and you may see these around Delaware or in other places in the country. These have become quite an important army of robots that we use really around the world in order to do wastewater-based epidemiology. So we'll talk a little bit about how this began and where it's going, as well as where we are today. So when we think about wastewater-based detection, it really began as a way to anonymously look for illicit drugs within a community or within a population. There was a really nice case study out of Israel in 2013 where they found polio. They identified polio virus in wastewater and then were able to start a vaccine campaign to be able to control that. And so there's some nice case studies that come out of this area of research, and we hope that what's been going on around the world in terms of COVID-19 is really one of those excellent case studies as well that people will be talking about in the future. So we think about what wastewater represents. It's not something that we typically think of, although we are very blessed in the United States to have an outstanding sanitation system. Really, our wastewater is equivalent to obtaining and analyzing a community-based urine and fecal sample. What's interesting, though, in terms of applying epidemiological tools to the analysis of wastewater is that we can think about these temporal changes that occur in microbial concentration, as well as diversity of different microbes that can be found. We might be able to also look for biomarkers and human metabolites. Variety of pharmaceutical agents also can be detected in wastewater, and that's a very exciting field of chemical analysis that continues to grow. This technology itself may allow for early detection of outbreaks, although we'll talk about why that shouldn't be seen as a standalone tool, but rather in collaboration with clinical testing. And if we design spatial sampling, we may be able to detect when an outbreak can occur or develop or spread across different locations. And of course, as we think about now where we're going with this technology, we're really looking at a COVID-19 disease, or SARS-CoV-2, the virus that causes COVID-19, but we're also thinking about where it goes in the future. So here's a summary of different substances that have been investigated using wastewater-based epidemiology in various countries around the world. And you can see there's a variety of different chemical agents on this slide. Opiates, the opioids there on the bottom next to the last row there, are really a common use of this technology and continue to be an area where people want to see growth, looking at illicit drug use, but it can be used for many things. One recent study that I was part of, actually, we were looking at caffeine and whether or not you could monitor human contamination in terms of E. coli levels, things like that in our water, by looking at caffeine levels, because we do partake in quite a bit of caffeine as a human population, and so that's also excreted and can be identified in wastewater. So there's lots of growing opportunities for this. A unique aspect of the wastewater-based epidemiology in terms of COVID-19 was really looking at the role that asymptomatic carriers might play in terms of transmission. And if you think back several months, this was a very big area of interest and an area that certainly is not new to clinicians or to the food industry. In my world, we often think about hepatitis A virus and norovirus, where you could be shedding after you've recovered, or in the case of hepatitis A, as shown here in this graph down on the left, you're actually shedding the virus before you have any idea that you're sick. And so those cases are really critical in terms of food transmission. But in terms of COVID, we are really still trying to grasp what does it mean to be an asymptomatic individual? How much do you shed? You know, when we think about the SARS-CoV-2 virus, there's certain things we're not going to get into today, but there are sort of this underlying premise. When we think about COVID as a respiratory infection, we have virus replication in our upper and lower respiratory system, but there's also replication going on in our gut. And how much does that play a role when we see changes in variants, and what's happening with asymptomatic carriers? So there's been quite a few questions, but we saw momentum of interest in wastewater-based detection when we were thinking about some of these questions about asymptomatic carriers. So even though we talk about the beginning of this with illicit drug use, there's always been a focus on virus detection. It's used historically to understand the burden of disease, the burden of potential for viral outbreaks. There are funded projects really globally and very important. We think about the different viruses around the world that are so critical in terms of diarrheal illnesses and respiratory infections. And much of this is done because the majority of disease with unknown etiology really are caused by viruses. And so wouldn't it be nice to have a technology that you can use to be able to detect the viruses in those communities? And again, looking at wastewater really represents a snapshot of what's going on in that community. Another aspect of this is that we really must have effective environmental surveillance. We have to have good researchers, we have to have skills and tools, and we need both field and laboratory resources. A nice case study, again, of this is what grew out of Pakistan. So Pakistan and Afghanistan are the countries that report ongoing transmission of indigenous wild polio virus. And in this case here in this preprint that's shown there in the middle of this slide, they were able in Pakistan to take the pipeline, literally the pipeline that was being used for detection of wastewater and looking for polio, and apply that to the SARS-CoV-2 virus. So really nice ways that laboratories can pivot and use this technology in novel cases. But it is still important in these areas in particular for looking for polio. So if we think about what happens to viruses in wastewater treatment plants, and this is an area that, you know, some people are are more involved in or understand more of than others. It's an area that we luckily take for granted, that we have this wonderful wastewater system. If you think about viruses there, starting on the left of this slide, with primary effluent is higher. That's where we're really going to find most of our viruses, in this primary effluent, much higher than even the secondary treatment. And that's where sedimentation occurs and viruses can attach to solids. They may interact with antagonistic bacteria or with protozoa, and then they get removed, and then they're usually inactivated by high temperatures. And so when it comes to the effluent, we really don't detect any SARS-CoV-2, and that's been very important for people to recognize that we don't find that. We, in my group, we were testing some effluent early on in the pandemic in the spring of 2020, and we did not find any virus there in the effluent, but did find quite a bit in the influence. And so I think it's good that we're all able to say that we're not finding it in the effluent. Also, I'll note here that SARS-CoV-2 is an enveloped virus and is not viable when we're detecting it in the wastewater. So we do go through a heat treatment, and I'll talk about that too, but we're able to be pretty certain that it's not transmissible or infective. That's different than something like norovirus or hepatitis A virus, which can survive during some of this system. So speaking of all of these different viruses, this table really provides you with a summary of the variety of human viruses that can be detected in wastewater. And there you even see some of the different levels of viruses that have been reported in terms of wastewater, and some can be quite high there, of course. We're looking at, in particular, global consideration. But there's a wide array of viruses. Some you'll note are waterborne, others are not, but have been detected in wastewater. And that's where science has its limitations. We don't quite understand the replication of some of these viruses in terms of fecal or waste transmission. The primary method for detection of these in wastewater really is PCR or quantitative PCR, and now the use of digital PCR platforms is becoming increasingly common, and that's advantageous because you don't need to run it against a set of standards, but you can actually get absolute quantification in wastewater. So the more we're using digital, I think the better we're going to be able to use it with this tool. And of course, you can see the coronaviruses here at the bottom, detected in excrement and detected in wastewater. So when we can't use PCR, or perhaps we're not quite sure what the target that we should be looking for in wastewater is, we can use metagenomic methods. And that really is looking at either shotgun sequencing or a wider approach to looking at what might be in wastewater. Again, this could be done if you're looking at specific temporal or spatial dynamics. Maybe you want to look at a location compared to another. You can see there's a variety of papers here, references where people have looked at wastewater and identified a whole array of different viruses and various families all in at one time. And so there's quite a lot going on inside our wastewater samples. Now, what's interesting about where we are from our past into how we transition into what we need and where we are in the present, we really think about how these methods are applied to different types of samples. Certainly, through COVID, we've seen a discrepancy in terms of how our communities face disease and the resources in which our communities are given. And so I think wastewater also allows us to look at some of those really critical questions. And these questions will remain important as we go forward, trying to understand resource allocation and how disease affects different communities. So we can use wastewater sampling to try to understand some of those dynamics, and with the appropriate clinical testing and laboratory data, we can also build these into really nice models that might be able to help us with future endemic disease. We'll touch a little bit more on that when we get to the future part of this. Another question that has remained really from the start of wastewater-based epidemiology in terms of viruses all the way through the present is what's the role of normalizing the data, and how do we use biomarkers to think about populations? Now, some of the leading scientists in this area are still uncertain about this. People have definitive thoughts on this, and some are really against trying to normalize the data. Others use various biomarkers or human genes or crassphage or something like that to be able to normalize this. And that will help us look at different communities, but we really need a solid scientific understanding before we do that, and that's still lacking. But here's a variety of some of the different biomarkers that have been used previously and are still being considered. So as we transition now from the past to the present, we need to really think about what do we do with this information, right? Science is pretty amazing in that we can continue to develop tools and be able to detect things that are going on in the environment, but how do we translate that into actionable data? And this is where we're still sort of struggling as we sit with a lot of information coming in terms of SARS-CoV-2, but we need to understand the comparison with clinical data. What does that mean given different populations? We need to be able to look at correlations between measured viral concentrations in wastewater and the reported clinical cases of disease. But in that comes that question about asymptomatic individuals. And so who is going for clinical testing? And as I think we all understand, there are limitations in terms of testing availability. So that is another layer that can confound some of the interpretation of this data, may perhaps make it less actionable. Then there's this validation, right? In the food world, with food industries, we must validate everything, right? We can't say to use a specific method until it's validated. So we want to be able to validate as well our prediction models, right? How do we know that we are accounting for the correct variabilities and that we are looking at the right range of concentrations of viruses? And again, more confounding information in terms of clinical data is that we really need to be able to track what's being used. We need to think about our communities and what types of indicators we might be using to tell that preventive measures were effective. So that brings us to the present, and I'm going to tell you a little bit about what's been going on in Delaware, in particular in Newcastle County, and also on the campus of University of Delaware. So this information has been used to support preventative measures. We've been able to work with Christiana Care actually during the flu season of last year to help understand how much SARS-CoV-2 is in the environment. With the residence halls on campus at University of Delaware, we've been able to work with Residence Life and be able to direct testing in some cases if a specific residence hall seems to have a higher number of cases all of a sudden, then those students in that dorm would be invited to go for testing so that we could try to catch individuals who were infected before they were spreading it to others. But we really, we need to think again about how this is actionable. It's very important that we compare this with clinical information. And so I want to make sure that we all understand that this is really a complementary approach with clinical testing, should not be used in standalone, especially during an active pandemic. So what was the response to these tools around the country? So Europe certainly picked up on this very quickly. This wastewater-based epidemiology was actually included in their discussion in October of 2020, and then also you can see here in their commission recommendation in March of 2021, they included really blatantly stating that wastewater testing should be complementary to the use of rapid antigen testing. And if you've been following some of the global news, you may have seen that Europe is doing these types of tests very well. They're widely available and used quite frequently, but wastewater monitoring is also used in conjunction with that. In November 2020, the World Health Organization also supported the use of surveillance of SARS-CoV-2 in wastewater, concluding here that can provide complementary and as well as independent information to public health authorities. So they went out a little bit more on a limb compared to what some of the countries are willing to do, in the US willing to do as well, but also stating specifically it's not a replacement for existing approaches. So it really is a good complementary tool. The U.S. was a little bit slower to adopt this. We had many public-private partnerships around the country who were very active in this wastewater testing. This organization here on the left-hand side, COVID-19wbec.org, has an incredible dashboard if you'd like to go look at that. They also have a fun Twitter feed. It's COVID Poops. And so it's really neat to keep track of what's going on all around the country. They've also published some papers themselves, and I'll show you their dashboard in just a minute. I really appreciate how the CDC responded to this, in that they were a little more cautious and now are working specifically with state agencies to make sure that this is being done in a consistent manner. And CDC certainly was cautious in terms of using this information in a response to decisions. So we saw universities embracing this technology and using it. We saw state and local levels using wastewater-based detection much earlier than we saw it as a national response. But now we do have a national response, and we have a national surveillance system, and I really think that that's going to be great to protecting us as we go forward. But you can see there are many factors that are involved in consideration of how this national system should work, right? We really need to have confirmation and confidence in the data, and it needs to be used with infection data where you are able to have testing in communities. And that's certainly not equivalent across the board, and it's not going to be used the same way across the board. And so that's some limiting factors in terms of actionable data that comes out of wastewater-based detection. So when we think about what's going on in terms of the actual laboratory work, this is an infographic from the CDC where they are showing a very basic way of looking for the virus in wastewater. So you have a sample preparation step. You concentrate the virus in the sample. You do an RNA extraction and then measure that. And then whether or not you use different biomarkers or normalize the data is variable. And then the SARS-CoV-2 data are presented as detected or not detected using the N gene, this nucleocapsid gene. There you can see them on the right-hand side, and either the N1 or N2 primer sets that yield positive results, amplification over a cycled threshold, and then we say that they are detected. And usually we report those as viral copies per liter, and it's important that we look at these as virus detected over time. And that's what you see here. Again, this comes from the CDC. And this is how they suggest that we look at reported wastewater data, where you have confirmed cases that are determined by clinical testing, and then that's compared to what was detected in the wastewater. Now, what's a little more, and still in the research phase, I would say, is this leading indicator analysis, right? And that really depends on a few things: how big your population is, so the size of your community, as well as how much testing is going on in the community. And that's really, really important. Another thing that's going to be, something that we should be considering as we're looking at this data overall is the timing of when we are taking samples and the frequency in which samples are collected. And I just want to mention here my personal opinion that I think we really, as scientists, we need to be careful that we don't misrepresent the data. I believe that this has been an issue across the COVID-19 pandemic. I always support education, and science literacy is really a critical piece these days. So we want to make sure that we don't overstep when we're talking about this data. We need to be able to make sure that we make clear differences in terms of virus detection and what that means in terms of public health actionable data. And I think that in the United States, companies that rushed to this early on really hurt their ability for using this as a national system. And I'm glad to see that we've caught up to that, but we do have to be careful to not overstep in terms of what the data might mean. So this can be an early warning system, which is extremely useful. It can help us to be able to stop an ongoing infection, depending on, again, the community size. And this study here by Bibi et al. That came out in July really shows that a modest lead time interval can range from 6 days for clinical testing to 4 days with wastewater-based epidemiology during community-level wastewater surveillance, where clinical testing is accessible. Though that 4 to 6 day window can really be incredible, but it has to be used with clinical testing. And as they found in this study, the potential lead time increases in settings with limited clinical testing. So the more testing you have, the better the window you might be able to detect it. This image I love, that came out quite early in terms of the work on wastewater-based epidemiology, really helped to organize some of the thought processes going on around the world. And here you can see this nice streamlined process of communication, action around the world, seeing that this is a public health emergency, and then pivoting some of the different tools and resources that are there to be able to collect wastewater, monitor wastewater, and then put that into epidemiological models and translate that to risk assessment. And this was done really well in multiple countries early on in 2020. Very exciting to watch the growth. I think you may remember, but I know many people in the United States were shocked when SARS-CoV-2 was found in sewage near Yosemite. Yes, right, if the virus doesn't care where it is, it's going to be able to be in a variety of different wastewater. And you can see it was here in Dubai, in places in Europe, in India. The Dutch scientists were certainly leading this effort with some of their incredible water research that they've been doing there. So it's really neat to see this grow and blossom around the country. This is the dashboard from that COVID-19 WBE collaborating center. What this group did, which is out of University of California in Merced, and they've won actually several awards for all of this work, is that they provided momentum and a platform for communication and for collaboration. And again, amazing to watch the numbers on this dashboard just grow and grow. So as of yesterday, they were at 2,691 different sites around the world. And that includes 93 published dashboards, 263 universities, and you can really see on all the continents where we're having this work done. And so it's really amazing to see the collaborative nature and discussion of science in this level. So here in Delaware, thanks to County Executive Matt Meyer, we were really an early adopter of wastewater-based epidemiology, and that was initiated through Biobot and then continued through University of Delaware. And it was, I think, Matt Meyer is really an outstanding politician as well as scientist. From my standpoint, where I've had a pleasure of speaking with him, where he's very interested in the science, and he was very interested in using this as a tool to complement clinical testing and really to protect the public health of Newcastle County. And so it was really wonderful to be brought in on this. And it was sort of a natural pivot for my lab group. This is Dr. Anderson-Coughlin, Brianna, who finished her PhD with me last year, and she had been studying irrigation water and various water reuse principles and places where they are forced now more and more, where we have less and less water to irrigate our agriculture with. And so we're looking at how do we safely reuse water. And one of those questions is, what is the type of viruses that are there? And so she had been looking at enteric viruses. And so we were ready and able to then pivot to testing for a different virus. And in doing that, we developed the Center for Environmental and Wastewater Epidemiological Research, which we fondly refer to as SEWR. And we were able to publish a couple papers on that, collect some of these amazing composite samplers with the help, again, of Newcastle County, as well as Duffield Associates. And so this was a wonderful example of a public-private partnership, all mounting together to be able to protect public health. This is the platform that we use. We're working with Kyogen Machinery, although there are many, many different companies that are available for this. We start off by incubating the wastewater sample at 60 degrees Celsius for 60 minutes. this will inactivate any of the pathogens that are there. So we also, in one of our publications, we show that this incubation step is very helpful at perhaps releasing some of those aggregates that might be holding in the virus. So we're able to detect even a more representative sample. Then we filter that sample, and again, then is concentrated. And this is actually a very small volume that we're using at this point, but it's a composite sample over 24 hours typically, although I'll tell you a little bit more about that as well. Then we do RNA extraction, followed by either a qPCR or a digital PCR. This is the dashboard for Newcastle County, and I think it's really a wonderful resource for people. I know many companies have been looking at it to see what's going on in the environment. Over time, you can see there on the right-hand side, the virus levels in the sewer system shown on the bottom really do reflect what's been in the clinical testing. And you see maybe more sharp peaks and declines in terms of the virus testing, and that's just where we were not detecting it or detecting it at much lower levels in the environment. And then, kind of following up, you see the clinical testing go back up. If you look at this over, it's a better glimpse of this over a long period of time from May through March of the following year, you can really see that very large spike that happened over the winter season, approaching the winter holidays of last year. Again, you see some of those more sharp decreases and increases in terms of the viral copies per liter that were detected in the sewer system. And it's, not uncommon necessarily to see that decrease. One thing that we're investigating with that is the dilution effects of rain, for example, and snowmelt, those kinds of things that we need to consider. The other aspect of looking at the data this way is that we really are looking for trend analysis. I think it's very important that we continue. We're not just looking at one pinpoint of data. We really need to look at the whole trend. There are many ways to represent this data. Here's just one of them. And as I mentioned, we worked with Christiana Care and other community members in Delaware to try to understand what was happening in certain populations. So there are 4 different wastewater treatment plants in Newcastle County, and that's what's represented here, as well as we take from various pump stations. And so we're able to understand what's feeding into these treatment plants in terms of overall amounts of virus. And here, again, everybody was trying to come up with a way to interpret this information. And by everybody, I mean really individual groups across the world. And so it's interesting to see how different people in different groups were reflecting that information and actionable or non-actionable data in terms of public health. And so here we're trying to look at some color coding where we're looking at the viral copies in wastewater. There you can see on the right-hand side the key for looking at various logs. And this is where the science is limiting, right? We have data to show that people who are infected with a high virus load, especially early on in the pandemic coming out of China, that they had high amounts of SARS-CoV-2 virus in their intestines and in fecal matter. And this really came from individuals who passed away, who succumbed to the disease. Most of this data comes out of China, and so we're really limited in understanding our shedding values. How much do people shed? Do the variants differ? And so we're trying to tease all of those pieces out as we continue to look at the data. The other part that's very important as we continue to use this as a tool is to understand catchment size. And so at the university level, we've been looking at fairly small populations in residence halls, and our wastewater treatment plants also represent that. So in treatment plant A, there we have our largest, that's the Wilmington area, so that's 500,000 individuals that's represented there. In B, that's Middletown, Odessa, and Townsend, and that's about 17,000 individuals. In C there, that's Delaware City, so that's less than 3,000 individuals. And then in D, that's Port Penn, and that has about 400 people. And so when you look at the changes and in fluctuations of this data, it really is going to be important to think about what's the size of that catchment, how meaningful are those changes, and if we're going to start instituting use of biomarkers, which we are continuing to study, haven't quite found the perfect one that we want to use for everything, and I think other laboratories are doing the same, and some scientists have really come out and said, it's not appropriate to use those, or it's difficult, continues to be difficult to really normalize the data. Here's some of the information that comes out of our university testing. So in the late fall semester, we started testing at 6 different sites, and we were looking for sites that would have the best flow, that had the highest population, because we did not have everyone back on campus last year, either in the fall or the spring, so very small populations of individuals. And flow is very important. Also, sampling location is going to be affected by where we can set up a sampler. Very difficult to collect samples from a manhole cover in the middle of a street, for example, that has its limitations. So there are many physical limitations to just collecting samples themselves. And I think at the top you can see some information there on our isolation buildings, as well as buildings that were used for quarantine. And so you can see there are some fairly high levels of virus. And so what we're trying to do is understand individuals who were put in isolation or quarantine at that time and try to understand the potential for the amount of shedding that could occur. On the lower level, we're looking at different residence halls. And you can see, again, various residence halls had higher virus shedding at times than others. And when that would happen, we would communicate quickly to residents' life, as well as the administration within the university, and ask them, share our information with them. They would ask questions that they need to, and then ask about when students might go in for testing, or how they might be able to set sort of an alarm in that residence hall and say, please make sure you are maintaining your distance, that you're wearing your masks properly, just to be able to try to convey to students the importance of maintaining their vigilance, so that we could try to reduce the chance of spread on campus. This also shows you a little bit more in terms of the trends of analysis in different residence halls. And in terms of community at Newark, we're really trying to understand what's happening in residence halls compared to what might be happening to students that are living off campus. And so on the right-hand side are maps that show some of the sewer systems where we're collecting samples for students outside of the residence halls that are also part of our University of Delaware community. Again, this is all done under the premise of trying to understand transmission. We're working with our colleagues in engineering who are also looking at students in terms of their behavioral patterns. And so we have models that are being built using this information, and again, trying to best understand how to work in terms of prevention and control going forward. There's a lot of questions that remain about sampling, and as more and more, even today, more and more small and mid-sized cities and universities are getting involved in this, trying to understand what types of samples should be taken. Can we take a grab sample at a specific time of day and that be just as good as a 24-hour composite in terms of being representative of how much virus might be there? Does the community size matter when it comes to that? How about the timing? And so on the right-hand side, this is a variety of studies that were done early on, really looking to see what types of influent should be sampled, could be sampled, and how does a grab sample compare to setting up one of these composite samples, which is wonderful to be able to have, but there are limitations on where we can place them for sure. We also were able to use these discrete samplers, which have 24 small bottles, and so we could collect samples from residence halls at different times of the day. And what we found, interestingly enough, is that between 1,800 and 2,400 hours, we found the most issues in sample collection. About 3 to 5 a.m. seems to be the best time to collect samples from students. Again, we're learning more and more about human behavior than perhaps we ever wanted to know. But this kind of information can be very important when it comes to identifying small catchments. So we're definitely making progress towards having a nationwide surveillance system. I think we're getting closer than where we've ever been before. This paper here that was published in Environmental Health Perspectives this year shows how we might use all of this information that I've been showing you today in this data stream that then could go to policy analysts. And this is an article from the Wall Street Journal that again talks about wastewater. And what's happening right now with Biobot, who, as I mentioned, started what we were doing here in Delaware, is that they have been funded really to look at nationwide wastewater. So various communities, including Wilmington, are sharing wastewater with them. They're doing their detection, and they're coming up with a nationwide spectrum here. They do normalize their data. They change how they normalize it with some frequency. So I'm not sure if they're normalizing it right now with a pepper mild model virus, for example, or a crassphage, but we're going to continue to hear and see more information on this. What about sequencing? How do we learn about variants when it comes to wastewater? Certainly, we can use wastewater to be able to identify variants, and there is more and more information on groups that are doing this. We are working a little bit on this, although we have not had the resources to put in as much effort as we had hoped, perhaps. And it's very tricky. And so it's kind of nice. We're sitting back, we're letting some of these other groups work out the kinks, and then we have some samples saved and we'll be able to use those. An exciting bit of evidence that's just come out recently, this is an October 15th publication online, that is showing the genome coverage. So what happens in these samples, you pull out wastewater samples and you can use tiling to be able to look across the genome to get as much sequence coverage as you can. And there, just by looking at the lengths of some of those bars on these two graphs, you can see they're increasing the ability of coverage. And that's exciting because wastewater is inherently a really complex matrix itself, but then your complexity goes up because you're looking across a community. And think of the different variants they have found in one individual. And so now we're looking at thousands of individuals. So it's pretty complicated. So it really remains as a way to be able to detect variants, not necessarily identify new variants. But we're continuing to make connections in these correlations. I think we're going to see more and more information coming out about what types of indicators we should use, human-specific biomarkers. We're studying several of these with my group, as are others, and so I think we'll see a lot of literature in the future on that. And so as we go into the future, where do we see this tool? Personally, I think it's so exciting to see these public-private partnerships around the world be reinvigorated over a very useful tool, right? In collaboration with technologies that are sort of old and existing, like looking through the pipes here with these tools that we were able to do on campus, to using really novel sequencing tools like nanopore technologies. I think we're getting closer and closer to global surveillance. And we're going to be able to use that to really help understand potential pandemics. This is a great project. This is the Global Water Pathogen Project, where they are looking at various surveillance technologies and techniques in terms of sewage. This is that same paper we looked at earlier from the environmental health perspective, but in this case, they're actually looking at predictions and preparedness when it comes to pandemics. So a model dashboard, for example, could look like this one on the left, where we have so much of the metadata, the exciting information about what's happening in terms of our populations. Here, can we address some of the inequities that we've seen in terms of our response to COVID-19? How can we change that so that we can treat people faster, better, more fairly? And then on the right-hand side, really looking at how so many variables play a role in understanding what that sample means, the frequency, the catchment size, the sampling method. Is there a way that we can get to a prediction model so that we don't end up with people being so sick? And of course, outside of our viruses, this is a great technology to be able to use for antimicrobial resistance. Again, we've had this invigoration of using this tool, and so there are more people some were already using this for antimicrobial resistance, but many others have now jumped on the bandwagon and are looking at antimicrobial resistance bacteria as well as genes in a variety of different samples. And here's an example of that type of a project going on around the world. And in my area, we're very concerned about an organism called Cyclospora cayetanensis that is transmitted by humans but causes important illnesses associated with fresh produce and also of unknown causes. So right now in the United States, on the right-hand side there, we have domestically acquired cases of cyclosporiasis from May through August. And there's over 1,000 illnesses, and the CDC doesn't know what food this is attributed to or how it's being transmitted. Sometimes we have direct linkage to salad mixes or mixed vegetable trays or fruit. Berries, for example, in history have been transmission vector. But now to use the pipelines that are in place, people are looking for Cyclospora cayetanensis in sewage. And of course, looking for a protozoa is different than looking for a virus. But it's wonderful to be able to see these pipelines in place and surveillance systems that can be used for so many other tools as well. So with that, I thank you very much for your time and your attention. And I do encourage you to look for wastewater data wherever you are. If you're traveling, it's kind of nice to see what's going on in that environment and help you with your own risk assessment.
Thank you so much, Dr. Kneil. This was just learned so much. Any question for Dr. Kneil?
I have a question. Can you hear me all right?
Yes.
So I'm just curious, when you're doing surveillance, like say in a college dorm, what do you ever run into any issues with like HIPAA at all? Like where suppose like everybody leaves for spring break, but there's like 2 students left in the dorm, and so effectively you're sampling from very small numbers of people. Does that ever become a problem?
That's a great question. So we have been really lucky to work with health services. And so they have the rights to be able to look at the testing data. We have not used any residence hall that had fewer than a few hundred okay. Students for sure. Part of that is for flow. We won't be able to collect the sample, but we are we only see anonymous information when it comes to the numbers of tests, and so all of that has been taken care of before we see anything. So again, that partnership is really important to try to make any good information, any meaningful conclusions out of the data.
Thanks.
I have a question. This is Levada Owens White, and I was wondering, with the using a preventative measure with including social distancing, would the advice or suggestion to close the lid on your bathroom to prevent that backflush, would that be helpful in preventing transmissions?
So it's definitely helpful in preventing transmission of things like norovirus or even salmonella or pathogenic E. coli, where some aerosols have been noted depending on the bathroom type, the flush type, the plumbing, those kinds of things. For SARS-CoV-2, I haven't seen any transmission specifically directed that way. There is this sort of famous outbreak that happened in Asia with SARS-1, where there was a backflow issue of the toilets, and so there was more flow and more aerosols that were produced. But under normal circumstances, it's probably a low risk, but definitely, it's not a bad thing to do, right? Close the lid, and then you don't have to worry about it.
But should that be included in our public health messages?
I think it's still low enough risk that it might take away from what we want people to really do to reduce transmission. If we know that it's so important that we physically distance, that we wear our masks, that we get vaccinated, I hate to add on something that is really of lower risk, nice to do, but not necessarily. We don't have the scientific data to support that.
Thank you.
I'm curious, and you may have said it, but does it take, between the data the collection and the result? How long does it take for you to provide the results?
So we try to do that as quickly as we can within 2 days, usually, barring instrumentation failure or other problems that can happen. But
so you are you are able to provide information to the public health
Yes.
organisms within 2 days. I mean, that's great.
Yeah, usually 2 to 3 days is the amount of time we give them. Now, but then you need people who will add the data to the dashboard in a timely manner. And as you get more and more people involved, it's, that can slow the process a little bit too.
Any other questions? Well, I still have questions. I can go on. So this nationwide effort, is it coordinated through the Department of Public Health all over the country, or how does it work?
Yes. So the CDC felt that it was most economical. It was the best strategy to work with state departments of public health. So yes. So in the state of Delaware, it does work through our Division of Public Health. And we've been really fortunate to be able to talk to them and talk about how do we do this on a statewide level, how do we continue partnerships and collaborations. So again, I think from that point of view too, it's been really nice to connect universities and private industries really across the country with their public health departments.
That's great. That's really fascinating. Any other questions?
Hi, Claudine. I have a question. Hi, this is Mina. Can you hear me?
Yes.
Okay, great. A great presentation. Thank you so much. I was wondering, well, obviously here in the US, we have more scientific ability and capacity, but in other countries, like developing countries, do they have the same resources to do such tests, especially during COVID?
Yeah, that's a great question. Actually, we've been talking to some colleagues in the informal markets in Africa, where they have an interest in testing for a variety of different agents. And in that case, so I showed pictures of these composite samplers, which have been wonderful for us to use. But there are many other ways to collect samples. One thing that is being further investigated or continues to be used in some locations is what's a modified Morse swab, where you can take a cheesecloth and put that into a PVC capsule, dump that into water, into a stream for a certain amount of time, pick that up, and then be able to collect the viruses from that and then process them. And in that point, it's sort of stable so that you can take time that you might need to send it to a laboratory. And so I do think it has the potential. People are interested in it. Now, exactly what kind of actionable data you might get from that is going to be variable depending on the location. But I do think it has a very useful aspect of it. And I really love it how in Pakistan they were able to be using the testing that they are doing for polio in their sewer systems or with sewage samples and then try to translate that to COVID. Again, it's difficult then to say, well, what should be an actionable level? And that's hard given variability in instrumentation. So I think as we learn more about whether or not you should normalize over a certain population, that will help us infer what we can do with that specific information. But it does seem to be being done. People are able to get samples to laboratories and get information that's useful. I think that's an incredibly important part of this whole thing. So thank you for that question.
Thank you.
And so I guess you I'm sorry, another question?
This is Laveda again. You mentioned that the composite sampling in urban areas is impractical, but I'm wondering, how are you utilizing community engagement to help with the research?
That's a great question. I would like us to do more of that. I would like us to be able to reach out a little bit more to specific communities. One thing I didn't talk about too much is our work with schools. So we are sampling in Newcastle County from middle schools and high schools and some elementary schools as well. And we are engaging the healthcare staff. We've talked to the nurses, we've talked to the administration, and we're getting students involved. So I think spreading the word about the science that's there, what we can find, how important our sewage system is for a variety of different aspects, at least from an educational perspective. We're trying to engage the community that way. I also think that following up with messaging about what this means and how when we see viruses increase in the environment in terms of sewage sampling, what does that mean to our own behavior? How do we use that messaging to try to protect ourselves and our society? So I think that this is, thank you for asking that, because I really think that we need more community engagement. I think we need everybody to be on board.
We do have some groups that are interested in the environment. I, in particular, represent our committee for the NAACP on environmental justice. So I'm very much interested in finding out how we could participate.
Oh, I'd love to talk to you about that. Perhaps we can follow up.
Thank you.
Thank you.
And also we have, within the CTR, the ACCEL CTR, we have a community engagement core. So, if that's something also, I'm sure that the core, and I wonder is also very involved in core, that, I'm sure there could be some partnership, with your research and our members within the core.
Elevate is a very active member of that core, so
Right.
I'll connect you, Levada.
Thank you.
Another question also. So you mentioned issues of flow. So I guess, now we see so much flooding, because of climate change. I guess that must be an enormous issue. And that's also a time where the community is extremely vulnerable, I guess, to this type of contamination. So how do you deal with these issues?
Yeah, that's a great point. So when thinking about how do we compare point-to-point data, the normalization, if we could look at how much of a human biomarker, for example, or a specific Bacteroides is an organism that we find in the guts of most people. If we're able to measure the amounts of those and compare them to the virus changes, we'd be able to account for dilution that occurs with flooding, for example. That's wonderful, but we're not quite there on the science to say that this is the best biomarker over another one. Certainly flooding in terms of environmental contamination is really critical. I think the good thing there is that SARS-CoV-2 is not going to be viable, but the other organisms many that come out of wastewater are, and so sewage runoff, sewage flooding, those are all very critical, especially in terms of food safety. And so there's a lot of education that goes into that, making sure that your systems are protected, you have buffer zones on your farms, those kinds of things that we talk a lot about those.
Right. Any other questions? Well, thank you so much again. This was really extremely informative and very, very interesting.
Thank you very much for the opportunity.
Thank you. Bye.
Bye.